Hi, and welcome to today's tour of MyCellHub. Let's start with a quick introduction. MyCellHub is a software as a service platform used for a large number of biomanufacturing related applications, including electronic batch recording, inventory and equipment management, cleanroom cleaning, and environmental monitoring. It's a cloud-based software with off-the-shelf yet entirely customizable modules. It's fully 21 CFR Part 11 compliant and specifically designed for GMP, boasting traceability features and audit trails, all based on electronic signatures and timestamps. There are three groups of benefits to using MyCellHub: preventing errors, simplifying regulatory compliance, and boosting productivity. The platform consists of two applications working and syncing in real time. The back office app, which I'm about to log into now, helps you manage resources and records, plan and assign workflows, review and approve processes, and generate reports. The companion app, which we'll demo in a few minutes from now, is mostly used in cleanrooms and provides your teams with interactive work instructions while collecting data in the background.
Hello everybody, thank you very much for joining us for our webinar, Automating Cell and Gene Therapy Manufacturing with Device Integration. In today's world, cell and gene therapy manufacturing is still often a very manual process. In order to scale in Pharma 4.0, we need technology like the Scinus bioreactor to automate these complex processes. These devices often generate a lot of data, and we don't want people running around with USB sticks to synchronize that data. At MyCellHub, we get more and more requests to synchronize data from any hardware with our software. So in today's webinar, we would like to address the benefits and challenges of such an approach. On today's agenda, we will first hear from Rens Roeslot, the Technology Manager at Scinus Cell Expansion, who will discuss their new bioreactor, Scinus NG, and its capabilities in monitoring critical parameters. Our second speaker is Teun Lambrechts, the co-founder of MyCellHub, who will discuss the work MyCellHub did with Scinus and the importance of device integration in cell and gene therapy manufacturing. We will then move on to a discussion where you will have the opportunity to ask your burning questions, share your thoughts, and engage with your peers about the exciting prospects of integrating hardware and software. As such, I will now give the floor to Rens Roeslot.
Thank you. I am Rens Roeslot. I'm the Technology Manager here at Scinus Cell Expansion. Scinus Cell Expansion aims to provide innovative technologies and processes to enable and enhance cell therapy and regenerative medicine. Traditional cell culture methods face challenges like manual handling, scalability, and the need to harvest clinical relevant cell numbers directly from open systems. Scinus addresses these challenges, offering a more efficient approach for cell cultivation. I would like to give you a short introduction to our new Scinus NG. It's a closed bioreactor system supporting GMP production. It has optimized control for cell culture conditions, and we aim to reduce the cost of labor. There are many applications for this system. For the adherent bag, for instance, we directly culture bone marrow derived stromal cells or direct culture of adipose derived cells, muscle progenitor cells. That's a difficult word. We also have a suspension bag that we, for instance, cultivate T cells or organoids, and there are many other applications. In short, the system consists of a bag cabinet, that's this side here, and that contains either an adherent bag or a suspension bag. This bag comes with pre-calibrated pH and DO sensors. The system has a unique way of rocking the cells, as you can see here, and the bag is expandable from approximately 100 ml to 1400 ml. Inside the system, there is a perfusion loop, and that controls pH and DO through the carboxygenator. On the other side of the system, there is a tubing cabinet, and that's used to do medium refreshments or sampling or anything else without disturbing the cells that are being cultured. On top of the system, you see a custom human-machine interface that's CFR Part 11 compliant. All data that the system is recording is stored on the device in a database. The database records DO, pH, pressure, handling, opening the door, all kinds of parameters. And our future development is focused on additional sensors that directly measure, or even soft sensors based on data. For this, we also have some collaborations like NextGen High-Tech for the integration of the Scinus within a high-tech cell culture platform with far-reaching control over cell culture process and also feedback loops. And with the Libra project for the development of a multi-sensing device that automatically monitors nutrients and pathogens. The system continually records the status of the system and the values of the sensors, and that's generating valuable data about the process. We also have a PC version of the software, and through that system you can retrieve the data in either a JSON file or a CSV file. Our system provides a big part of the whole process, but it will be part of a bigger chain. And we see the need for our customers to incorporate the data from our system to their system and vice versa. Thank you.
Thank you very much, friends, for sharing such a detailed overview of the exciting work going on at Scinus Cell Expansion. Like you said, devices like bioreactors, don't work in a vacuum and need to integrate with other softwares, considering all the data that's being generated. The interactions between the operators, the device, and the data generated by the device need to be recorded properly. And this is where MyCellHub can play a key role. So now we will hear from Tone and his experience with device integration in cell and gene therapy manufacturing.
Thanks, Clara. Happy to introduce myself to you guys. MyCellHub is a software platform for the management of GMP data. Our goal is to get rid of every single piece of paper that you might use in and around the clean room, all while remaining GMP compliant, obviously. We do this with two different apps. And as you can see on the slide on the left, we have our back office application where you can manage all the resources in your facility. This ranges from the bills of materials of your processes or how to set up the bioreactor, for example, to the batch records of the manufacturing, to cleaning tasks and the users and their trainings, for example. And then on the right, you have what we call the companion app. This is a mobile application where operators get interactive work instructions, and we can help them via the tablet to collect the right data at the right time while the operators are executing the work. And we basically use different configurations of these two apps to digitalize every single workflow that you might find in a GMP facility, and that today is still often done on paper, for example. Think of batch records with inventory tracking and so on. On the next slide, I won't bore you too much with all the modules and the extensions of MyCellHub. Today, the focus should be on the device integrations. And basically, the MyCellHub device integration, it allows you to connect hardware, like the bioreactor, or any other type of device that you might have in your clean room, and to connect that with the MyCellHub software. And by using this connection, you can use the data of the device directly in the electronic batch record. Typical examples are bioreactors, but also cell counters, or maybe even label printers, I think, are very typical examples that you will find in almost any ATMP lab or clean room. And often the goal of the device integration is just to get the device data into the batch record and centralize data from multiple different devices into one central location, so it's easier to manage it. But a nice thing, since MyCellHub can also send information to a device, we can also operate those devices remotely, which provides some benefits in terms of not having to go into the lab. You can basically operate the device from your office or where you're sitting at home. So on the next slide, you see what we did together with Scinus on a Eurostars project, where we integrated the Scinus bioreactor data into the MyCellHub electronic batch record. On this video, you see, for example, a demo of an operator who is changing the set point of the rocker speed in the Scinus bioreactor on the MyCellHub tablet app. And when the operator is saying send to the tablet, you will see that in the background, this rocker speed will change from fast to slow or something like this. And this is just one example. A manufacturing recipe would typically consist of a large combination of these set points that you send to the bioreactor and receive. But basically, you can build your own recipes with this. And this means that you don't have to manually enter set points in a device. You can actually manage your recipes from the tablet. And the operator also doesn't have to come in a lab or in the clean room to just press some buttons, let's say. The operator can do this from anywhere. And so maybe as a summary slide, I would say that it's awesome to start automating your ATMP manufacturing, but by adding this additional layer of device integrations, you can lay the foundations for way more efficiency gains. So, for example, this device integration helps to centralize the data from multiple devices, and this data can be linked automatically to the right manufacturing lot, for example, in your documentation, which basically helps you tremendously to facilitate the traceability and the reporting of your operations. We can also make sure that the batch records are even more interactive by providing situation-specific work instructions on how to interact or how to operate the device, for example, but also by making sure that the device set points or the recipes basically of the devices are an integral part of the batch record. It's much more simple to manage different recipes from one central location instead of, for example, having to upload a recipe or sometimes even different versions of a recipe to many different devices. And since you have access to all the device data in real time, it's also easier to keep an eye on your process just by watching the pH and DO measures fluctuating, for example. But in the next phase, and Rens also hinted towards that, is that we can make more intelligent use, I would say, of that data to design, for example, real-time process control strategies. And by using the digital processes and this additional data, we will be able to reduce even more the manufacturing errors. We will also be able to streamline the quality compliance and probably also provide data-driven optimizations for manufacturing processes in the future. So I hand it back to you, Claire.
Okay, great. Thank you very much for your presentations, Rens and Tone. And now it's time for the Q&A session. So it's up to our listeners to ask the questions. And we have our first question, which is directed for both Tone and Rens. And it is asking, could you discuss with us how the collaboration process happened and what were the steps to get to this integration?
Shall I start, Rens?
Yeah.
All right. Well, this project was done in a bigger Eurostars project with some other partners, but how we started to work together was basically to map out all the data, the different data points that the Scinus bioreactor was able to collect and send out. Rens helped us a lot with mapping all those data points, also the minimum and maximum values, and how those values are communicated. And then for us, it was a matter of mapping those data points into the, in this case, OPC, UA, communication layer with MyCellHub. And basically then it's defining again in MyCellHub which data points need to map where. So it's a lot of data mapping, and I think those are the three basic steps. So understanding which data is there in the device, then setting up the communication, and then in the MyCellHub software, mapping again to the MyCellHub concepts, which data points need to be stored where.
And Rens, on your end?
Yeah, from our side, it's a challenge for us to find out what customers want the machine to do at a certain point. So we thought about, do we want to integrate some kind of batch record or user settings in the machine, and it's very complicated to do so. And with the integration of MyCellHub, that moves the power to the customer to define what their process needs and directly control our machine and other machines. So that's a huge value. Then that's something really hard for us ourselves to do. So this is a great collaboration.
Great, thank you very much. We have Giacomo with a few questions coming in. The first being, how you showed integration with Scinus bioreactor. I think, Tone, this is directed towards you, in order to monitor and control it. Any other real-life examples of integrations with multiple instruments other than Scinus? Example, you cited the cell counters.
There's multiple. Scinus was the first project we did, so it was a bit of a learning curve as well. But once actually we have now this communication layer, it's not too difficult to add multiple devices. There are integrations with the nucleocounter. There are integrations with multiple label printers, for example, at the moment. And there's some other integrations in the pipeline, which I cannot yet really disclose, but yeah, there's multiple.
Okay, great. And I think that kind of works well with Giacomo's next question, which is, and can you devise some more details on how MyCellHub could practically help organizations of work in a facility, manufacturing of batches, release, logistics, reagent warehousing with this?
Yeah, of course. It's a bit off topic, I think, but the MyCellHub software is basically a collection of modules where we help manufacturers with batch recording and everything that feeds into the batch record. For example, the logistics is not really our strong point, but what we do is the from, let's say, the material reception to all the material release workflows, to then bringing in all the materials in the clean room via a picking workflow. Then once you're in the clean room, we help digitalizing the manufacturing batch record. Basically, all the in-process controls can be tracked, and then all the more downstream, let's say, data analytics or samples can be analyzed and recorded on the tablet or on the computer. And then basically once, let's say, the manufacturing run is over, you can generate with a single click of a button the whole batch record, basically. So really our goal is to get rid of every single piece of paper in the lab and the clean room. So it's a modular approach with some process building tools as well. So basically any workflow that you're currently doing on paper, I'm convinced that we can digitalize it as well.
Okay, great. Now, Giacomo, thanks you for addressing the questions. Rens, we have a question from Elizabeth. She says, Rens, you mentioned the integration of multiple sensors. The combination of more sensors and the centralized collection of data opens up opportunities for data analytics. Could you elaborate on this, please? Thank you.
So now it's more or less a closed box in which, yeah, we record the data from a run and we store that finally with a run, and we see that as the total batch record from that process. What we now might see in the future, that collecting the data and collecting more data in the future for let's say glucose or other parameters that you might can think of, and combine those into smarter solutions for your whole process. So you could think of that if you see a certain parameter shifting or moving, that is an indication, oh, this is your time point to start thinking about cell expansion, or this is time to refresh media, or those kind of steps that you can distill from the data based on multiple runs you have in storage, kind of to say so.
Okay, great. Thank you for that explanation, Rens. The next question is directed towards both of you, where so there is much talk about decentralized manufacturing, which promises to bring down the cost of ATMP manufacturing. How can Scinus and how can MyCellHub contribute to this in the future?
Can you answer that, Toon?
Sure, I can try. Well, the decentralized manufacturing, I think on the bioreactor, it's quite clear, right? You want to have a controlled process. For example, I don't know what you exactly mean with the decentralized, but I'm imagining that there is some hospital that is manufacturing, or many different hospitals manufacturing the same product, or at least some CDMO that is manufacturing on multiple locations. You basically want the bioreactor to make sure that your whole process is how do you say, replicatable in all the different environments, and this bioreactor helps there because everything is automated and it basically does the job every time the same. So that's on the decentralization of the manufacturing. I think that the problem with, or the challenge with decentralizing the manufacturing for MyCellHub is that actually you want to centralize all the data, because now you have many locations where the same process is running, but let's say as the central manufacturer, you want to have control over all the data. You want to be able to say that, okay, on location A, what we manufacture is the exact same as on location B. You want to have control of the raw material inputs. You want to have control of over the QC readouts. And so basically centralizing the data from decentralized manufacturing is one of the bigger challenges that I see. And MyCellHub can help there. We're a cloud application, for example, just to start with. You can log in from anywhere, and basically everyone that has that login, or every device that has access to the MyCellHub platform, can see the same database. So that is one of the main advantages, I think.
Yeah, and I guess it's also about removing data silos, you know, in the era of Pharma 4.0, trying to decentralize manufacturing but centralize data. You need these type of device integrations to remove all these data silos that we've created in the past decade. And that's how both the, you know, device integration is really a key, you know, point moving forward. Now, Rens, did you want to add to that?
No, I don't think I have anything to add other than I see the necessity. This is a data silo. It just records data on its own, but I think we can benefit if we can put in data from other devices or other processes that can enhance the cell culture.
Yep, definitely. Now, for Tone, we have a question that asks, can you discuss how device integration can improve the efficiency of the manufacturing process? Can you give an example on this?
Sure. I think we had briefly some examples, but it's it all starts like Hans is sitting in the lab now. To go there, well, the lab is where you work, Hans, is still quite open, but imagine you have to go into a clean room, you have to gown, you have to go into the clean room, you have to press the buttons on the device physically, and then basically go back out of the clean room often. Just practically logging in from your office maybe, or even at home, just logging in on the device, being able to check the set points and maybe changing some of the set points, that immediately a big time saver, but also depends on what type of clean room you have, obviously, but also reduction of contamination risk, for example, but also on the process itself. Often, if you now want to have access, let's say, to the dissolved oxygen concentration from the device, you have to physically go there. But basically, if you want to have the data and link it to your batch record, you somehow need to grab the data from the device. While if you would have an integrated device and software, electronic batch record and device integration, that data can be linked automatically to the manufacturing run, and you don't even have to think about collecting the DO data between two different time points or something. The data is just there, and it's automatically linked to the right bioreactor, to the right run, to the right manufacturing lot. So I think the time efficiencies are clear. And on top of that, all the, let's say, traceability features, or enhanced traceability from which device comes which data, from which manufacturing run, all that metadata is better integrated, and it just helps you with documenting everything in an efficient way.
Yeah, thank you for that, Tone. Now we have the question coming from David, who asks, to ensure an adequate device integration, what are the key features to consider for the device and for the software? I guess that's a question for both of you. Rens, would you like to start?
It's kind of a difficult question, I think.
Yeah.
Is the question, well, I'm thinking of the regulation then, or like,
Yeah.
For example, what we worked hard on is basically this audit trail of the device. You need to have on every action that you do with the device, or the device sends data to the software, we need to have this completely integrated audit trail so that every change on the data is completely traceable. So at least on the software side, that is a big requirement. Of course, there's so many requirements. The connectivity, you need a proper connection, sure. Especially if it's wireless. What else? There's quite some requirements. Maybe the question is a bit too generic.
Yeah. Rens, did you want to add any more specific features to consider?
Yeah, I mean, our system is Part 11 compliant, and I think that's also for MyCellHub, and that's a really important step. Things to consider might be if you want to use patient data or medical information. I'm not sure how you should deal with that. That's something I'm not familiar with it, so that might complicate things. Other than that, I think that's maybe a question for the whole field. What kind of standards do we want to use? Is it OPC, or is it something else, or I don't know. But it should all be able to talk to each other, and that you could circumvent that by using MyCellHub, and device A talks something else, and device B, but they both talk to MyCellHub, so then you can solve that. So that's something to consider. What devices you choose, and can they interact with each other?
Yeah.
Yeah, I think that standardization is an important, critical point for our industry now. Yeah. Every device, at least from MyCellHub side, it's a new integration project. Of course, once it's done, you can implement it at other sites easily. But the first setup is always a bit of a tailor-made setup in this case, and that's something that would be nice to streamline. And maybe one other requirement that I remember we struggled a bit with, Rens, well, from my MyCellHub side, was the buffering. Your device can send a lot of data, and somehow you need to be able to, even if, I don't know, the internet connection is gone for a couple of seconds, or if there's just too much information to be sent over your Wi-Fi connection or whatever, that buffering of the data, that is something that's also a big requirement that you think either. I think, Rens, your device now has an internal hard disk as well. So that on the hardware side, you can play around with it, or you can mitigate it. And on the software, we also have some tricks to make sure that data is not lost if there's too much data.
Yeah. Now we're logging data every 3 seconds in this machine, so you generate a lot of data. Yeah, definitely.
Yep. We have one last question, and it says, how long does it take to integrate a new instrument to MyCellHub?
Depends a little bit. So here the standards come into play. Let's say it's a new bioreactor. There's quite some different data streams, different units, different frequencies, stuff like this. There's some time of mapping out like what is there, and then mapping it again to our software or our communication layer. If everything is nicely documented and well structured, the first proof of concept can be done in a week. Of course, then validating everything and checking all the, let's say all the, how do you say this in English? All the edge cases and so on, and stress testing, it might take a bit longer, but it typically doesn't take longer than 1-2 weeks to do a first integration.
Okay, great. Thank you for that information. I guess one of the main points that has come through this webinar is the need for collaboration amongst, all the stakeholders in the cell and gene therapy industry. And once we standardize those processes and work together, then, we can work towards a more, efficient manufacturing. Now, this is the end of the webinar. I'd like to thank Rens. Thank you very much, and Tone, for your time today. And also, I'd like to thank in particular all the listeners that joined in. Thank you for your questions. Please feel free to reach out to either Tones, Rens, or myself via LinkedIn or our websites on Scinus.com and MyCellHub.com. I hope that you have gained some valuable insights and that this webinar has given you some tools and ideas in following through with your digitalization journey and kickstarting your hardware and software integration. So thank you very much and have a great day. Thomas, I see you've sent a question. Yes, you will receive the recording link. Okay, bye everybody. Thank you.
Thank you.
Bye.